Let's talk now about the adaptive immune system. And big difference between the adaptive immune system and the innate one is that the adaptive can learn. This is one difference. Another difference that it has memory. It has memory, immunological memory. What does it mean? It means that it can learn how to defend the body against even new viruses, even new bacteria sometimes. So it can learn how to defend that, okay? And then it records the infections. So it has a history, think of. It has a log that records what happened. So that is for the adaptive immune system. A third difference with the adaptive immune system that it can recognize. It has self-recognition. What does it mean? It can differentiate between self and non-self cells. So this one can detect the non-self cells, the cells that they don't belong to the body. Now, the thing about this adaptive immune system, we said we have mainly two types of cells. We have the B cells and we have the T cells. Those are the two major types, the B lymphocytes and the T lymphocytes. The B lymphocytes, their way of action is something called the humoral immunity. What does it mean? It means that they release antibodies into the blood, and those antibodies, they attack the infected cell. Okay, so sometimes the B cell is, think for example in the tonsils, in the mouth, and it's secreting antibodies to attack a viral infection in lungs, for example. Okay, so this is in the mouth or in the throat, and it sends those to the cells of the lung. So the B cell, it doesn't have to come near the infection area. So this is humoral immunity. The T cells, we call it cellular immunity. Cellular immunity. Why? Because the T cells need to come in contact with the infected cells. So they need to establish a form of connection with them. And we'll try to understand that. Okay? They need to recognize, technically this approach is to recognize the infected cell. That's why we call it cellular immunity. This is only for the T cells, whereas the humoral, it's only for the B cells. Now, both of them would need antigens to be activated. So both the B cells and the T cells need to recognize the antigens. And for those antigens, they need to know if this is a self or non-self antigen. What does it mean self? It means it belongs to body. Like for example, liver cell. In your own body, your own liver, those liver cells are self cells. They are displaying self antigens. Versus non-self doesn't belong to body. Like what? Like wrong blood transfusion. This is one example. Another example, cancer cells, okay? Or infected cells with viruses. So all of those cells will show in their service non-self antigen, and therefore we will call them non-self cells. So what is really the antigen? Any part of the protein, sometimes it's a protein, sometimes it's just chemical structure that can, be recognized by the T or the B cells. And the same protein or the same antigen can be recognized by different antibodies or by different receptors. And this is based on the 3D conformation between, the area of recognition and the protein in this case, or the antigen. So for example, think of it like a key and lock. If there is match, then there is recognition. If there is no match, then there is no recognition. But this protein or this key or this antigen, not necessarily that, if it doesn't match, it's a save. Think of it like this. You have this antigen, and this is, let's say, non-self, and we can recognize this part, this part, this part, this part, this part. All of those can be recognized as places for non-self or as the places for recognition. So for example, like here, it needs an arm to recognize it like this. This arm cannot recognize this area, for example, or cannot recognize this area, for example. It doesn't mean that this area is okay. Doesn't mean that this area is okay and this area is not okay. Sometimes cells will recognize only one part of the antigen. The cell that recognizes this part not necessarily is able to recognize this part. So that is for the antigens. And those antigens, we will respond to them usually by antibodies. So usually antigens could be on the surface of the cell or could be floating, but the antibodies also can be on the surface of the cell or floating. Those antibodies will be able to recognize the different antigens. So it's always like a 3D conformation between the antibodies and the antigens. So for those antigens, we said both B cells and T cells need antigens to get activated for activation. However, there is a big difference. B cells can recognize antigens by their own. What does it mean? It means that the B cell can examine or investigate the antigen by itself. It doesn't need any help. However, the T cells cannot recognize the antigen by themselves. Therefore, the T cells need antigen presented presenting cell. What does this do? It presents, from the name, presents the antigen to the T cells. It presents the antigen to the T cells. Not only presenting it, but presenting it in certain way as well. Not only showing it, but showing it in a certain way. Those antigen presenting cells could be macrophages, could be dendritic cells, sometimes even B cells. Those cells, the way they are presenting the antigen, they have to display it on something called major histocompatibility complex, MHC. Major histocompatibility complex, or MHC. And this MHC has two classes, MHC-I and MHC-II. MHC-I presented on all nucleated cells of the body. MHC-II presented on only the antigen presenting cells. If we want to draw this, we said for the adaptive immune system, we have the B cells, T cells. Those can recognize antigens directly, cannot recognize directly. Therefore, in need for antigen presenting cell, or ABC. Those antigen presenting cells, we said from their name, they present the antigen to T cells. Why? For activation. Those antigen presenting cells could be macrophages, could be dendritic cells, could be even B cells. All of them can be antigen presenting cells. Now the issue is, for those antigen presenting cells, not only presenting the antigen, but they have to must present on MHC. They must show it on the major histocompatibility complex. For this MHC, we have two types, class 1 and class 2. Class 1 in all nucleated cells, here only on antigen presenting cells. Let's do some drawing. Let's say this is a liver cell. On its surface, there is the MHC-I. This is the MHC-I. And over there, it will display this antigen. So the T cell would come and recognize the antigen, and it has to recognize also the MHC-I. This is the T cell. We'll see some details regarding that. But if this was a macrophage, then it has MHC-I and it has MHC-II. This is MHC-I, but also it has MHC-II. Both of them can display antigens. And again, the T cell can come and bind to this, recognize this, and recognize this, or it can bind this and recognize this. We'll see more details about this, what's happening here. But that is, just to show you the difference between MHC-I and II. Now here, why this macrophages has both MHC-I and MHC-II? Because it's, first of all, it's nucleated. So if it's nucleated, it must have MHC-I. And then, because it's antigen presenting cell, it has II. Think of this like a cop or policeman or woman. Those police members or personals, they will have normal driving license, let's say, or normal ID, like the one for the public. And then they have the special ID that will say that I am a policeman or a policewoman. Okay, so that is kind of the same thing here. MHC-I is the normal ID for all nucleated cells of the body. The special ID is the MHC-II. This one says that I'm antigen presenting cell. Okay, so I will stop here. In the following video, we will start talking about how the T cells and the B cells get activated, and we will see some details about all of that.